Open the chat. Phylogeny and systematics. So far we've looked at various aspects of evolution. Starting with Darwin, we looked at Darwin's discoveries of all these different organisms, animals, plants, fungi, especially beetles. He was inordinately fond of beetles, I think, Darwin, and collected them and brought them back to Europe, where the knowledge of organisms around the world greatly expanded in an enormous way. And of course, he also suggested that species weren't fixed, but rather changed over time, which meant there had to be ancestral species. And indeed, species had gone extinct that didn't quite make it for one reason or another. And so that took us to the origin of species, which was about speciation, which was about how one species can change to another, and how the accumulation of changes can lead to macroevolution or really big differences, divergences, such as reptiles from birds, for example. And then looking at the tree of life and how all this information is coordinated, that was about different areas that provide evidence for evolution by natural selection and speciation, including the kinds of geologic events that opened up, surprise, surprise, new ecological opportunities. So when the earth froze over, for example, snowball earth, that killed a lot of stuff. But there was still a few that survived because they were super hardy, you know, on the ice. And then when everything melted again, there was all these opportunities. There was nothing already occupying the ecological niches that were around. They were all aquatic, of course, all in the ocean. And so then these organisms, this small unicellular ones, started to form colonies. And we talked about sponges in the beginning of term when we looked at invertebrates. And I think one thing I love the most about that film, The First Life by David Attenborough, was showing the sponge. Because I remember suggesting that you could put a sponge in a blender and blend it, separate all the cells, put the cells in a bowl of water, and the sponge would reform. And hey, he verified that, David Attenborough himself. So that was really cool to see. Somebody that they did it, of course, through a syringe. So they broke off a little piece of sponge and they separated the cells through a sieve. And then lo and behold, the sponge formed again, which is a very interesting idea of a colony forming before a multicellular organism, which has more organization. So what I'd like to look at now is how we organize things. So this is basically about organization. Phylogeny is building phylogenetic trees, which are relationships between extant species, so those are the ones alive today, and common ancestors. This field has really come such an amazingly long way. So systematics is placing organisms in groups, being systematic about the whole thing. So phylogeny is the evolutionary history of a species or a group of related species. And how do we get it? We've seen lots of ways, including the ones that David Attenborough likes to talk about a lot, is fossils. So he had to look at boxes and boxes of fossils. And the fossil record can show ancestral characteristics that may have been lost over time. So let's see, what are some of the characteristics that were lost over time? So many species have gone extinct. I think that the fractal organization was lost with that one group of animals, but it's resurfaced. So there is still some fractal organization of organisms. So that's what we can look at. We can look at the fossil record for data to try to create a phylogenetic tree. We can look at morphological data. What do animals look like? We can look at biochemical data. How similar are their proteins? We can look at molecular comparisons. How similar is their DNA? So biochemical, we're usually looking at proteins. Molecular, we're looking at DNA similarities. Morphological, of course, just what somebody looks like, their phenotypes. So systematics is a very analytical approach to understanding diversity in relationships. It's quite mathematical also. It can be very mathematical. We're not going to get into the math too much. So systematics, what are the relationships? Well, one thing that has been revealed over time by combining all of these various lines of evidence, including genetics and DNA, shows that humans and fungi are more closely related than humans and flowers, interestingly. I don't know that to what exactly that is due to, but it has something to do with the arrangement of genetics and cell biology, the biology of the cells themselves. And when you see two extant species like this fungus and this human together, and this is the branches where you find them, they're related, and their common ancestor is here. That's how you read a phylogenetic tree. And these two are more closely related than either of these are to the flower. But the flower is related. It's just that the common ancestor was farther back. So it's kind of like if you were to suppress this into your own pedigree, you know, this is your grandparents, your parents, and the kids. So you are less related to your grandparents than your parents. As a matter of fact, you're 25% related to your grandparents, 50% related to your parents. And so we can use probabilities like that when we build phylogenetic trees, but on a larger scale than a family. So sedimentary fossils are quite common. Attenborough is always talking about marine environments, and anybody you'll read, like Bill Bryson, for example, who wrote a history of nearly everything. It turns out that the fossils that we have are almost all marine, probably 95%. Why? Because that's where sedimentation happens. So sediments settle on ocean floors. They are eroded from land. They are eroded from land and washed to sea. And then, of course, they're washed to sea. All these sediments form, they get really heavy. Eventually it becomes rock. And then the plates move and volcanoes happen. The rock melts, volcanoes happen. And some of that ocean floor is pushed way up, and often to the tops of the mountains that you see today. That was one of the big questions way back. You know, people were finding fossils at the tops of mountains. It's like, WTF, how can you have a fossil here that is a marine organism? It's the top of a mountain. Well, now we know about plate tectonics, and we know that when plates come together, they crumple and they form mountains. Yeah. So there's loads of different kinds of fossils. Here's some bones. So bones fossilize fairly readily. They're minerals after all. This is a petrified tree. So one, bones, two, petrification. And that's when the tissue of the tree is replaced with minerals, but retains the shape of the tree itself. Um, Here's a leaf fossil. This shows carbon tracings. So the leaf decomposed, but the carbon was left over because presumably it was squashed between maybe, I don't know, two pieces of rock or in the mud, I suppose. But the carbon remains, and carbon is black, so you can see it quite readily over a light background. Very interesting. These are casts of ammonites. They're about 375 million years old. So 4 casts. So what those are is the animal gets stuck in sediment. The sediment makes a mold around the animal as it hardens. The animal decomposes, but that mold is replaced with minerals. And that's what a cast is. What else do we have? Oh, here's insects. So amber is something that forms from the resin of trees, and it hardens into amber. And insects can be caught in it, and they're preserved then for as long as the amber lasts. What's another one? I think this one down here is 150 million year old dinosaur tracks. So I think those are called trace fossils. Which are traces left from the animal's presence, and these are footprints. Quite enormous footprints, as it turns out. And you know, people like Leakey, for example, who looked at the first humans, traces of the first humans in Africa, and could tell by looking at the traces that they walked upright. So somebody who's an expert in that field can look at things like footprints and even a tail like swooshing along the ground and be able to tell what kind of animal that might have been or what kind of movement that animal portrayed. Very interesting field. And then this is presumably a mammoth, 23,000-year-old mammoth, frozen. Well, that's another way. And of course, bogs. Bogs are a good place to petrify something or fossilize something. Yeah, so you can also be confused when you're building your phylogenetic tree by convergent evolution. You have to be careful if you're using morphology, because these are animals, one from Australia, one from North America. Their ancestor is quite ancient, more ancient than you would think. You might look at them and go, well, they must be very closely related because they have such a similar morphology. Be like, well, you'd be arguing with another researcher. No, I think they're very similarly related. And they'd be going, well, I don't think so, because one is an Australian, one's a North American. You'd be like, but look at what they look like. You might be having this argument with another scientist. But anyway, what you'd have to look at are different features. So in Australia, for example, this one is marsupial, and this one is placental. That's tricky because you might not see it in, well, you might be able to see it in fossils, I guess, but somebody who is an expert could probably tell whether an animal was a placental or a marsupial. But anyway, that has to do with their reproduction, you know. And these are the adaptations that they have. Look at this. This is just like the star-nosed mole. This one is very similar to the star-nosed mole, actually. So they have those front legs for burrowing, and they have incredible noses because they're looking for food underground. So they have very similar features, but they have an ancestor that's quite distant. So how do we evaluate molecular homology? So homologous features, we looked at that before. We looked at the radius of the arm, which can be found in bats, in whales, and what else did we look at? Cats and humans. So that's a homologous structure. But you can also look at homologous molecules, basically looking at DNA segments from different organisms. How similar are they? So homologous DNA segments are here. So these are both the same. And then there were mutations to this DNA. So that here is a deletion, and here is an insertion. But the homologies still remain. But they're not aligned anymore. But somebody who studies molecular DNA through practice and practice and practice can tell that those homologies still exist in the current DNA. They use computer programs to realign. So that's also used to build phylogenetic trees, the evolutionary relationships and ancestry of organisms. So just another repeat of hierarchical classification that Linnaeus developed, binomial nomenclature. The domain here is Eukarya, kingdom Animalia, phylum Chordata, class Mammalia, order Carnivora, family Felidae, genus Panthera, and the species is Pantheropardus. So we can link this hierarchical classification of phylogeny to phylogeny, because you would expect the species that are in this genus. Oopsie, no, that's not the genus. This whole thing is a species. This is the species. This is the genus. The species that are in this genus, Panthera, well, they'll have a lot of similarities. And because all these species are similar, you would expect that their ancestor would have Panthera qualities that was passed on to diverging species. So there's the Panthera genus, and etc. along the way. Let me show you what that looks like. Oh, I like this. I forgot I put this in here. It's a pickup line. Your name must be Phylum because you're above class. You can use that. I saw one the other day too. It was so funny. It was like, oh yeah, oh yeah. Are you a parking ticket? You must be because you've got fine written all over you. Yeah, feel free to use that too. Okay. Anyway, moving along. So this is what I mean by that. Here's a phylogenetic tree and branching. So these two organisms here, these two animals are in these genuses. Mephitis and Lutra. This is a skunk and this is an otter. They're basically in the weasel group. So their common ancestor must have had the features of a weasel. So the family is Mustelidae. And what else? So Panthera are in the genus, the genus Panthera is in the family Felidae. The canis, which are your dogs, are in the family Canidae. They have some similarities. Cats and dogs both have canines. That's a feature of carnivores. They both have penis bones. That's a feature of carnivores. I can even show you one. Because I was teaching reproduction to the other class. And I like to shock them with this right here, which is a baculum, which is the penis bone of a walrus. And, you know, its adaptive significance is that it's difficult to get to an egg because eggs must be protected except from the best males. So that's partly why. And also reproduction sometimes has to take place very quickly. And so the penis bone helps with that. Also, vaginas have evolved to be more difficult to penetrate in animals, and the penis bone helps with that too. So there's lots of reasons for a penis bone. Adaptive reasons. And it turns out all carnivora have penis bones. That's the order. But also, of course, canine teeth and different digestive systems than herbivores. So canine teeth, penis bone. Baculum is the word for that. And yet, Canidae, canine teeth, and the baculum as well. Phylogeny, canine teeth, and a baculum. But they have a common ancestor back here that was a carnivora, but they diverged. So when you're looking at the genus level, they're separated, partly because the cats have retractable claws. I'm sort of simplifying it quite a bit, and the dogs do not, non-retractable claws. So that causes them to diverge. Okay. So each branch point represents divergence of two species. So this is the common ancestor back here somewhere. Never mind, it's already written there. I'll just write it again. But deeper branch points represent greater amounts of divergence. So a wolf is more dissimilar to a domestic cat than another cat-like animal is. That's really all we're saying. We're starting to develop a method for building a phylogenetic tree, and we're using characteristics to build that tree. In other words, we're using morphology here. But we could use a lot of different methods like DNA. So we construct a phylogenetic tree based on shared characteristics. And when I went to university, it was all about phylogenetic trees, and then this whole new aspect of building trees came along. I used to argue with my friend Rick because I didn't find it that much more useful than the other way of building a phylogenetic tree, but it's really similar. It's called cladistics. And it shows patterns of shared characteristics among taxa. Taxa are any individuals that are in families, for example, or a taxa might be a genus. So this system groups clades within which what is called a cladogram. So a true clade is a group of species that includes an ancestral species and all of its descendants. And I can show you what that looks like in a diagram. And this is known as cladistics. the study of resemblances among clades. Is it easy? I don't find it easy, but my friend Rick found it really easy. And then, you know, it was because I studied with Rick that I got like really great marks in a couple of these classes. But it's an interesting field, and we're going to use it. So you can nest a clade within larger clades, but not all groupings are qualified as clades. So a valid clade is what's known as monophyletic. So here's a valid clade right here. It's this ancestor and all of that ancestor's descendants. That's a clade, a true clade. A paraphyletic clade is not really a true clade. It includes some, but not all of the descendants. And this is all in the eyes of the observer. So this tree is already built. We're just looking at different groupings. So there you go. That's a paraphyletic clade. There is another one, isn't there? Yeah, polyphyletic grouping. That includes a number of organisms, these ones and these ones, but they lack the common ancestor. So you have to go quite far back. Basically, because you're skipping this one, you can no longer call it an actual clade. It fails the cladistic test. So we're going to do some cladistic analysis. When I say we, I mean you. You just need to know a couple of terms. One is a shared derived character. That's a novelty, an evolutionary novelty that's unique to a clade. This is where it gets complicated and why I don't particularly like it, but nevertheless, we're going to learn it because that's the way people do things these days. But a shared primitive character is a homologous structure that predates the branching of a clade from other members. And this is where we had the arguments, because really the other members that you select are fairly arbitrary. It's shared beyond the taxon we are trying to define. It's basically just wording. So it's called outgroup comparison. So you're finding your, you're finding your shared primitive character with an outgroup. So this outgroup has one character that its descendants have as well. But it's difficult to differentiate, but to differentiate between them, you have to use an outgroup. An outgroup is a species or group of species closely related to the ingroup, which is the species that we're studying. So we decide as researchers, we're going to study this group of species. And we need to find an outgroup which is closely related. And then we make what's known as an outgroup comparison. And it assumes that the homologies between the outgroup and the ingroup must be primitive characters that predate the divergence of both groups from a common ancestor. I know, it's crazy. But this is what I'd like us to do. It's quite interesting. I think you'll enjoy it. You will do an outgroup comparison. So we can focus on characters that are derived at various branch points in the evolution of a clade. So here's a character table. So I'll get you to build a character table in this exercise. And it shows the similarities, the characters that are shared. So here we have the taxa that we're looking at. We're looking at a lancelet, a lamprey, a tuna, a salamander, a turtle, and a leopard up there. And these characters that we're looking at are the backbone, the jaw, the forelegs, the amniotic shell, and hair. So does the lancelet have hair? No. Does the lamprey? No. Does the tuna? No. The salamander? No. The turtle? No. The leopard? Yeah. So the leopard has hair. That's terrific. What about the shelled egg? Who's got that? Well, the turtle does. The turtle lays a shelled egg, and so does the leopard. And we start to build by characteristics the relationships between these organisms. And then we can build our cladogram. So this is a cladogram. And I know it seems like gobbledygook at the moment, but as soon as you see this exercise, it will start to make a lot of sense. Yeah. So this is our cladogram. This is our outgroup is the lancelet because it's the least it's the least what sort of looking for related, but it has the shared character that lancelet have of all chordates. It's got the chordate features basically. And then the advent of a vertebral column, the lamprey, hinged jaws, the tuna, 4 walking legs, the salamander, an amniotic egg, the turtle, and then finally hair, the leopard. So when we're looking at a tree like this or building a tree like this, we are getting closer and closer to more and more shared characteristics. So let me stop there and I'll get you started on the exercise.